This is a model of DNA. In DNA, there are many different parts. The first part is the double helix. And the double helix contains the nucleotide, its basic structural unit and building block for DNA. They contain phosphate, deoxyribose sugar, and nitrogenous.
And that's the nucleotide.
The phosphate joins together the nucleotides in the DNA
Sequence.
The phosphate is the white strands
that connects with the nucleotide and brings them together. The next part to this is the deoxyribose sugar, which is the blue and black strands right here. The last part is the nitrogenous bases, which are adenine, guanine, cytosine, and thymine, which are the beads that you see on this model. And all of these things are held together by hydrogen to make. This is the cell cycle. In the cell cycle, it comes in 4 different phases, but in 3 different checkpoints.
The first phase is growth 1, also known as G1. It's where the cell grows and the organelles duplicate.
Then you will reach checkpoint 1. The second phase is synthesis, which is when it has 46 chromosomes and is duplicated to 92. And then you will reach checkpoint 2.
Once you reach checkpoint 2, G1, also known as growth 2, growth checks the 4 arrows and mistakes.
And then you will reach checkpoint 3.
Oh my bad.
Then you will reach the last stage, which is when the cell divides in mitosis, in the last stage. The next thing is the DNA helicase, which is the enzyme that unwinds the double helix by breaking the hydrogen bonds between the complementary bases, which is basically just a cutter of DNA that goes in between the nucleotides. The next thing we're going to talk about is polymerases. The DNA polymerases are enzymes that create DNA molecules by assembling nucleotides, which is the building blocks of DNA. So basically, this is just the glue that repairs the DNA. This is a picture of everything that we just explained to you. This is the DNA helicase cutting into the DNA and separating everything to create the new DNA. Right here is when the polymerases is being created to go into the new DNA. And on each side, there are now 2 DNA molecules.